This week's lesson is on transcription and translation of DNA. Last week we talked about DNA replication, which is how DNA copies itself, and how it uses RNA then to go through the processes of transcription and translation. First two questions on your assignment are the definitions for transcription and translation. To transcribe doesn't only apply to science, it also applies to any time that you copy something. So in biology, to transcribe means to copy using the same DNA language of nitrogen bases, either A, T, C, G, or A, U, and C, and G. So when things are transcribed, it's the same language, it's talking about DNA and RNA, and it's talking about the code that those two molecules have. Translation, on the other hand, just like if you were to take something in Spanish and translate it into English, or something in German and translate it into Spanish, translation means to go between two different languages. The end product of transcription and translation is for your cell to make proteins. So to translate means to go from the DNA, RNA language of A, T, C, and G into a different language called the amino acid language. And amino acids are made of codons, or sets of three. And these sets of three then turn into the proteins that your body uses. Let's talk about protein structure. Proteins. Remember we talked earlier that these are macromolecules. They are very large as far as molecules go, unlike things like water or oxygen gas or carbon dioxide. Proteins are made up of amino acids. Those are the subunits for a protein, the smaller parts. A polypeptide is a string of amino acids. Proteins are made in chains or strings. And there are 20 possible amino acids. These are arranged in different orders to make the variety of proteins that your body needs. Proteins are assembled or they're made on a ribosome. The ribosome is like the factory floor where the proteins are made. So the questions we're answering today are, how do we get from the bases found in DNA, like A, T, C, and G, to the amino acids? And how do we get from a bunch of amino acids into actual proteins? Which codons code for which amino acids? The genetic code, I know this is another question on your assignment. The genetic code is the inventory or the list of linkages between nucleotide triplets. Nucleotides, remember, are A's, T's, C's, and G's, and the amino acids that they code for in any organism. A gene is a segment, a piece of DNA that transcribes a segment of RNA. So a gene is one piece of the code, and it turns into a piece of RNA. And a codon is a series of 3 RNA bases that make a specific amino acid. 3 RNA bases make one specific amino acid. And remember, there are only 20 possible amino acids. Here is a review of replication. DNA, the arrow here means that DNA makes a copy of itself. In this process, the double helix or the ladder unwinds, it untwists. DNA is now single-stranded because the ladder gets split in half. And then a new DNA strand forms using the complementary base pair rules of A, T, C, and G. A goes with T, C goes with G. And then you have 2 copies of the same DNA. Why does DNA replicate? DNA replicates in order to prepare for cell division. Remember, when the cell divides, the DNA has to make a copy of itself so that both new cells get the same genetic information. In this way, your whole genome, or all the DNA that your cell has, gets copied. The next step after DNA replicates is transcription and translation. And This is an overview of what that is. This is sometimes referred to as the central dogma. You'll see this again in the second assignment for this week. When you go from DNA to RNA, that is called transcription. You are transcribing. It's in the same language. We still use the A, T, C, G, or U if you're going into RNA. However, when you go from RNA to a protein, that's different languages. So that is called translation, like going from Spanish to English. Remember that both DNA and RNA contain a sugar, a phosphate, and a base. The sugar and the phosphate form the outside part of the ladder, and the bases form the rungs. However, DNA and RNA are not exactly the same. DNA is double-stranded. It has two sides to the ladder. RNA only ever has one side. DNA is formed with a deoxyribose sugar, but RNA is formed with a ribose. And the bases for DNA are C and G, A and T, cytosine, guanine, adenine, and thymine. But the bases for RNA are C, G, A, and U, uracil, instead of thymine. Now let's get into transcription. This is when DNA turns into RNA. It's the same language. We talked a little bit about this last week at the end of the replication PowerPoint. So here you see the DNA has unwound. You've got complementary DNA strands. And the RNA gets built onto one of those strands. Actually, it gets built onto both of those, but it's only showing one. The RNA forms base pairs with the DNA. Remember that instead of a T, the RNA will build its base pair with a U. However, if the DNA has a T, then the RNA will still use an A. C with T, A with U. There is no T, there is no thymine in the RNA strand. The primary transcript is the length of RNA that results from the process of transcription. So in this diagram, the primary transcript is anything in this area. If your DNA strand looked like this, and the DNA was going through the process of transcription, what would the RNA strand look like? If you started with an A, a C, and a G, and notice how they're broken into groups of 3, that's important. If you started with an A, C, and G, what would the complementary bases be in RNA? We know that A can't bond with T because there is no T. So it would have to be A with U, C with G, and G with C. In the next set of 3, ATA would become U, A, U. Next, CCC, that's fairly easy, becomes GGG. And TGA would become A, C, U. You, on your assignment, need to complete the last 2 transcriptions. What would the RNA look like if the DNA strand had CGA and then GCG? Let's now talk about the major players in transcription, meaning what are the different molecules needed for transcription to occur? The major one, of course, is mRNA. There are different kinds of RNA. This is M. The M stands for messenger. This type of RNA takes the coded information for the making of proteins and carries it to the ribosome from the nucleus. It actually travels. It's a messenger. The other major player is RNA polymerase. This is a complex of enzymes with 2 functions. They have 2 jobs. First, they unwind the DNA sequence, and then they produce the primary transcript. The definition of primary transcript, again, was the length of RNA that comes from the DNA. And they do this by stringing together the chain of RNA nucleotides. The nucleotides are the bases. So RNA polymerase is an enzyme that has these two jobs. The processing of messenger RNA. The primary transcript is not considered mature mRNA. It's got some things in it that don't need to be there. The DNA sequence has coding regions, called exons, and non-coding regions, called introns. So in order for the mRNA to be considered mature, it has to go through an extra processing step. It has to, something has to remove the introns. These are the non-coding regions, the things that basically don't have, they don't produce any amino acids. They have to be removed before the primary transcript is considered mRNA and can leave the nucleus. So to make mature mRNA, you can see here, this is the primary transcript, which means it's a piece of RNA. And genes, it says at the bottom, genes are made of parts represented in the mRNA, the exons, and parts that are transcribed but not present in the mRNA, the introns. Introns are removed from the primary transcript and exons are spliced together to make mRNA. So the thing that does this, basically what it's saying is we don't need these purple parts. They don't serve any function as far as an actual code goes. So we have some enzymes that loop those, splice them out, cut them out, and then the messenger RNA gets attached together right there. And then the messenger RNA, the code is completed and it can go out to the nucleus, or out of the nucleus to the ribosomes to be made into a protein. Now that transcription is done, it went through, we've got the messenger RNA, we've made it mature by taking out the pieces that we don't need. What happens next? We now have mature mRNA transcribed from the cell's DNA. It's leaving the nucleus through a nuclear pore, just a hole in the nucleus. Once it's in the cytoplasm, it finds a ribosome so that translation can begin. We know how mRNA is made, but how do we read that code? How does it actually turn into a protein? That happens through a process called translation. Remember, translation is different languages. This goes from RNA to protein. And it's the second stage of protein production. mRNA is on a ribosome. In this picture, the ribosome is these two purple pieces. The red and yellow, blue and green, that is your protein. Very long, very complex structure. This here is your mRNA strand, which has the code. And this here is the complementary piece that gives you the amino acids that make the protein. Just a quick thing about ribosomes, they have two pieces. They're separate in the cytoplasm until they join to begin translation. So it's kind of like a top and a bottom half. In the one assignment it talked about, it looks like a right hand with a thumb. It's got a large piece and a small piece, and it contains 3 binding sites. The E, the P, and the A, which you're not going to need to know. The P is what we're most concerned with because that's where the codons actually attach. In translation, this is, again, the second stage of protein production. The mRNA is on a ribosome. That's represented by this code here. Now we have the second kind of RNA, tRNA. T stands for transfer. And the tRNA brings the amino acid to the ribosome. This is what tRNA looks like. You can see it kind of looks like an upside down T. It's called transfer RNA. It's bound to one amino acid on one end. That would be this. These turn into the protein. You link a bunch of these amino acids together and you get your protein. And the anticodon on the other end complements or matches the mRNA codon. So you have an anticodon on the transfer RNA and a codon on the messenger RNA. And a codon is simply a set of 3 bases in a row. The job of tRNA is to keep the amino acids in the correct order for the protein to function. The tRNA lines these amino acids up using the mRNA code. So just to review, you've got 3 kinds of RNA, and I know this is a question on your assignment. The first is messenger RNA. This is found in the nucleus, and it will migrate or travel out to the ribosome. Its job is to carry the DNA sequence information to the ribosomes. You've got tRNA or transfer RNA. This is found in the cytoplasm, and its job is to provide the linkage between the messenger RNA and the amino acid. It transfers the amino acid to the ribosome. The amino acid would be found at this end, and the anticodon would be found at this end. And the last kind is the ribosomal RNA. This is also found in the cytoplasm, and it's simply a structural component of the ribosome. So how does the DNA code actually get read? Every 3 DNA bases pair with 3 mRNA bases. That's called the codon. Every group of 3 mRNA bases encodes a single amino acid. The codon is the coding triplet of mRNA bases. 1 codon will equal 1 amino acid. So here's how this looks. In the nucleus, you have DNA. Transcription happens, and you get a strand of mRNA, messenger RNA. And each set of 3 is called a codon. When translation happens, you have these codons get read, like a code, and it brings in the correct amino acids. Those amino acids get linked up. These green boxes are amino acids, and when you link them together, you get a protein. There are some special codons. There's one codon that always starts the process. This codon is AUG. This codes for the amino acid called methionine, or MET. Methionine is always the beginning of any protein. There are 3 codons that stop this process: UAA, UAG, or UGA. Any 3 of those show up, and you know the protein is at the end. The genetic code says that each triplet is the code for an amino acid, 1 amino acid. A string of amino acids makes a protein. These are, and I said at the beginning that there were only 20. These are the 20 combinations of amino acids that you could have in order to make a protein. This one, AUG, this is your start codon, and these 3 are your stop codons. The abbreviations here, these represent the amino acids that get made by each set of 3. A review of transcription versus translation. Transcription. The process by which genetic information encoded in DNA is copied onto messenger RNA. So going from DNA language to RNA language, which is the same language. Translation is the process by which information encoded in mRNA is used to assemble a protein at a ribosome. Translation goes from 2 different languages, the language of bases to the language of amino acids. Transcription occurs in the nucleus. Translation occurs on a ribosome. Transcription goes from DNA to messenger RNA. Translation goes from messenger RNA to protein. As always, if you have any questions on this, I know these processes seem complicated. If you have any questions, please let me know.